DNA transcription. Please note that I am not a professional educator. In the last two videos, we learned about the history and structure of DNA. But now it's time to wonder, how does DNA become you, or a plant? Well, DNA gets read. In biology, we say that DNA gets transcribed. In this video, I will tell you about DNA transcription, how DNA is initially read. First off, we have to start with the eukaryotic cell, more specifically, its nucleus. There are lots of other organelles in every eukaryotic cell, but we don't really have to worry about them yet. In the nucleus, there is chromatin, or squiggly DNA, and there's also some RNA, but we'll talk about that later. Here's one strand of DNA in the typical double helix form, but I'm going to show it as a simple ladder, because that's easier to manipulate. First off, there's a set of nucleotides on the DNA called a promoter. A promoter is a set of nucleotides that calls a specific enzyme. In this case, that enzyme is RNA polymerase. So, the promoter calls an RNA polymerase, which comes over to the DNA and unzips it. But what is an RNA polymerase? Is the word erase a clue? It sounds plausible, but actually no. The real word parts are polymer, or a larger molecule made up of many smaller ones, and ase, which means enzyme. Therefore, an RNA polymerase is the enzyme that builds the RNA polymer. But what's RNA? The simple answer is that it's single-stranded DNA, but that doesn't really explain everything. There are four other key differences. One, it's much shorter than DNA. It only contains a small amount of the DNA message. Two, it has a different purpose from DNA. Instead of storing the genetic code indefinitely, it just carries it. We'll look into this later. 3, its 5-carbon sugar is ribose, not deoxyribose. Yeah, that's right, we're getting really nitpicky up here in Ganondosville. No, but really, instead of deoxyribose in DNA nucleotides, each RNA nucleotide has a ribose sugar, a fancy schmancy and slightly more oxygenated sugar. Wait, why'd thymine just change to a U? Well, that would be the fourth difference, uracil. Instead of thymine in DNA nucleotides, RNA nucleotides have uracil. All the other bases are the same, though. An important thing to remember is that thymine and uracil are not complementary, like cytosine and guanine, but equivalent, like cytosine and, well, cytosine. So now we know a bit more about RNA, but what happens next? Well, the RNA polymerase just draws from the local resources, in this case, free-floating RNA nucleotides. Inside the polymerase, these nucleotides quickly rearrange themselves, forming hydrogen bonds with their complements on the DNA strand. More importantly, they've formed covalent bonds between themselves, becoming an RNA strand. This process is repeated many times, and in fact, the RNA polymerase moves along the DNA strand, unzipping and rezipping as it goes. But eventually, once a full gene is copied, the RNA strand exits the RNA polymerase. The polymerase itself also packs up and leaves. Now, the DNA strand is whole again. But what happened to the newly created RNA? It actually left the nucleus of the cell through a nuclear pore, braving the wateriness of the cytoplasm, with its journey to be continued. Nucleotide, nucleotide, nucleotide. Actually, we'll learn what those mean too.